Today topic of discussion is humoral and cell mediated immune response. First we want to know what is immune response. Um, the immune response is how your body recognizes and defends itself against bacteria, virus, and substance that appear foreign and harmful. So, immune response is how your body recognizes and defends itself against the bacteria, virus, substances that appear foreign and harmful. Immune system protect the body from harmful substances. So it protect our body from harmful substances by recognizing and responding. How it will do? By recognizing and responding to antigens. Antigens are substance, usually protein. Actually it is a protein on the surface of the cell. Antigens are substances, usually protein. It is presented on the surface of the cells. Virus, fungi, and bacteria, non-living substance like toxic chemical drugs, foreign particles also considered as antigen. Microorganism and non-living substance like toxin chemical drugs and foreign particles considered as antigen. The immune system recognize and destroyed or try to destroy substance that contain antigens. So our immune system recognizing and try to destroying or destroyed that substance containing antigens. Next, types. Two type of immune response. One is humoral immune response. Second one is cell mediated. Humoral immune response, B cell involved. Uh, Cell mediated immune response, cell T cells only involved. Now we will discuss how to will form B lymphocytes and T lymphocytes. All blood cells, okay, all blood cells formed in hematopoietic stem cells in bone marrow. So this is the bone marrow. Here hematopoietic stem cell is presented. All blood cells formed in hematopoietic stem cells. Then B precursor cells formed, then mature B cell formed, then it will mixed with the bloodstream. So hematopoietic stem cells, P precursor cell, matured B cells, then it will mixed with the bloodstream. Like this only hematopoietic stem cell, T precursor cell, then it will enter to thymus. Thymus is an immune organ, then it divided into T helper cells and T killer cells. Then it also mixed with the bloodstream. First, we will discuss that humoral immune response. Whenever foreign particles, we consider that one is the antigen. It may be microorganism or any substances, toxins, chemical drugs, any particles. It will enter to human body. Our immune response will activate. So, first that foreign particles first enter to macrophages. This one is the macrophages. Macrophages will help now that processing started. That time that MHC2 complex, major histocompatibility complex 2, will come out, then it producing antigen peptide. It producing this MHC2 complex will producing antigen peptide. When this antigen peptide will come out, T helper cell will stimulate. T helper cell will stimulate. T helper cells have TCR, that means it having that receptor. T helper cell having receptor. With the help of receptor, it will accept that antigen peptide. These macrophages releasing antigen peptide with the help of T cell receptor, it will accept this antigen peptide. T helper cells releasing cytokine. This time it will releasing cytokines, especially that interleukin 2, T helper cells will activate T helper 2 cells. T helper cells will activate T helper 2 cells. So foreign particles enter to human body. Macrophages will help that antigen. Then processing will start at that time. Major histocompatibility complex 2 will form. Then it will producing antigen peptide. That time the T cell stimulate, T cell having T cell receptor, it will accept that antigen peptide. Now it will releasing that cytokine leukotriene 2. That time it will activate T helper 2 cells. So foreign particles are antigen entered to human body. Macrophages inhale antigen, then started processing with the help of NHC2 complex, it releasing antigen peptide. T helper cells stimulate. T helper cells also having that receptor, it will accept, it will sharing that antigen peptide. T helper cells now releasing cytokine extractly that interleukin 2. Now it will activate T helper 2 cells. First T helper 0 cell, then it will activate T helper 2 cells. Simultaneously, on the other hand, B cells also having that receptor. Whenever antigen will enter, that processing will started. So now B cell also producing antigen peptide. It also producing antigen peptide. Already activated T helper 2 cells will accepting this antigen peptide. So B cell having receptor, B cell start to processing, it also producing antigen peptide. Already activated T helper 2 cells releasing interleukin 3. T cells, it also having receptor, T cell receptor, it will share B cell producing antigen peptide by the release of interleukin 3, IL-3. After B cell processing, B cell produced two type of cells. So B cell here activated, it also producing antigen peptide. T already activated T helper 2 cell will sharing this antigen peptide. B cells producing two type of cell, B memory cells and activated B cells. This activated B cell producing lot of cytokines. These activated B cells produce plasma cell. This plasma cell producing antibodies. Here immunoglobulin. 5 different type of immunoglobulin: IgG, IgA, IgM, IgE and IgD. In this case of interleukin 4, 5, 6 released means it producing IgG antibodies in plasma cell. This one is the common one. When this interleukin 4 and 13 released means it producing IgE antibodies. This one is whenever allergic reaction, that time this production is high. Then interleukin 5 and 10, it released means it producing IgA antibodies. It fight against if bacteria is presented in respiratory and digestive tract. This IgA antibody is also called as secretory immunoglobulin because it is secreted by mucous membrane. So after B cell production, two type of cell production will occur: memory cell and activated B cell. It is also called as copy of B cell production. That activated B cell producing plasma cell producing antibodies, 5 different type of immunoglobulin. Interleukin 4, 5, 6 released means production of IgG. This one is the common. Then interleukin 4 and 13 means IgE antibody. It will whenever allergic reaction, that time this production is high. Then interleukin 5 and 10 released means immunoglobulin A. This one also called as secretory immunoglobulin because it is secreted by mucous membrane. This one fight against bacteria to presented in respiratory tract. All immunoglobulin enter stomach through digestive tract. After that it will destroyed except IgA, because this IgA having J chain and secretory potency. Not this one, because of J chain and secretory potency, it not affected by gastric juice. Next level, all immunoglobulin will attach to mast cell, because mast cell having receptor Fc fragment. After that it will secreted histamine. So through digestive tract it will reach to stomach. After that it will destroyed except IgA. Then next level, all the immunoglobulin will attach to mast cell. So overall humoral immune response, this one here, whenever foreign particles will come, macrophages inhale that foreign particles. Now it started processing. That time MHC2 complex will come out, then it producing antigen peptide. When this antigen peptide will come out, T helper cell activated. It stimulate it with the help of T cell receptor. It will accept antigen peptide. Then it will releasing cytokine interleukin 2. Then T helper 0 cells will activate T helper 2 cells. Simultaneously B cells activated. B cells having B cell receptor, it also producing that antigen peptide. It will attach to it will share that antigen peptide to already activated T helper cells. Now these activated B cells producing three type of immunoglobulin: IgG, IgA, IgE. Overall, this humoral immune response, it will considered as immediate immune response, because whenever foreign particles enter to human body, it will react faster. After that it will causes inflammation. That means it will causes organ swelling, pain and fever. This is all about humoral immune response. Again, one is cell-mediated immune response. Here, the destruction of infected cell by cytotoxic T cell. Destruction of infected cell by cytotoxic T cell, or destruction of intracellular pathogen by macrophages. Two way of destruction. Destruction of infected cell by cytotoxic T cell, or destruction of intracellular pathogen by macrophages. Here no antibody involvement. Here activation of macrophages and NK cell, natural killer cell. No antibody involvement. Activation of macrophages and NK cell. Here production of antigen specific cytotoxic T lymphocytes and released various cytokines. Here production of antigen specific cytotoxic T lymphocytes and releasing various cytokines. Here foreign particles in our body, okay, not from outside. Here cancer cell, macrophages inhale. Now that process will started. MHC2, okay, will come out. After that it will releasing antigen peptide. On the other hand, that T helper cell start to stimulate. Here this T helper cell having receptor, this receptor will accept this antigen peptide. Okay, after that it will activate T helper 1 cell with the help of cytokine interleukin 2. Now this T helper 1 cell with the help of cytokine, it producing three component. In the case of interleukin 12 and gamma interferon means released, means it producing NK cell. This NK cell directly kill cancer cell. Interferon gamma released means it will activate many macrophages. These macrophages will activate T helper 0 cell. This T helper 0 cell will activate T helper 1 cell. So it is a continuous cyclic process. Macrophages, T helper 0 cell, T helper 1 cell. It is a cyclic process. In the case of this interleukin 12 and gamma interferon released means it producing T killer cell. Okay, this T killer cell, T helper cell and T killer cell. What is the difference? T helper cells mediate all immune response. It have CD4 complex. C that T killer cell, it having CD8 complex directly destroyed tumor cell. This T killer cell directly destroyed this tumor cell, cancer cell, with the help of this T cell receptor. T killer cell share that antigen peptide to this target cell, cancer cell. Okay, after that, this antigen peptide having two chemical information, two enzyme also presented, granzyme and perforin. This antigen peptide having chemical information how to kill that cancer cell. So by using granzyme, it make a hole. After that, it will send perforin. After that, it will kill that cancer cell. These are the way this chemical mediated immune response will cell mediated immune response will kill that cancer cell. So foreign particles in human body, it inhaled by macrophages. Now process started. MHC2 complex come out and producing antigen peptide. That time T helper cell stimulate by using T cell receptor antigen peptide. It release cytokine interleukin 2. It will activate T helper 1. Then T helper 1 with the help of cytokine producing 3 component: NK cell, macrophages, T helper cells, T killer cell. These T killer cells producing two type of enzyme, granzyme and perforin. It will make a hole. It will after that it will reach to this perforin reach to cancer cell. It will kill cancer cell. Thanks for watching.